Hello, my name is Karen Nuñez and I'm a PhD student at the International Laboratory for Human Genome Research from the National Autonomous University of Mexico. The title of my work is Logical Modeling of Dendritic Cells in Vitro Differentiation from Human Monocytes, Unravels Novel Transcriptional Regulatory Interaction. This work was led by Dr. Alejandra Medina and Dr. Dendritic. Dendritic cells belong to the immune system. They are antigen-presenting cells, and once they are mature, they can activate naive T lymphocytes, and in this way trigger adaptive immune response. Dendritic cells can be obtained from the hematopoietic stem cell and also can be differentiated from the monocyte. And this differentiation can replicate it in the laboratory. When we obtain the monocyte from the bloodstream and incubate it with interleukin-4, the result is a monocyte stimulated. When we incubate the monocyte with CSF2, this leads to the differentiation of the macrophage. When we incubate the monocyte with the combinatory of interleukin-4 and CSF2, this leads to the differentiation of the dendritic cell. CSF2 and interleukin-4 activates 4 signaling cascades: MAP kinase, P3K, NFKB, and JAK-STAT. This leads to the differentiation and dendritic cell development. The aim of our work was to study and characterize the regulatory mechanisms that control dendritic cell differentiation, and integrate this information in a dynamic model in order to complete the regulatory network. The methodology that we follow was to search into the literature information regarding human monocyte to dendritic cell differentiation, and the differentiation were done in the same condition, and to have at least one reference per interaction to complete our gene network. We perform simulation and model predictions, then we compare with experiments, and we realized that we needed to add new regulatory interactions. To do this, we use epigenomic data to add to the model and complete the circle. The first model that we construct based on literature has 2 inputs, CSF2 and interleukin-4, that are the green nodes. Then we have the 4 signaling cascades, and in the yellow nodes are the transcription factors. And as we can see, there are few target genes. So we wanted to know which genes were regulated by those transcription factors. We select specific genes to each cell type, for the monocyte, the macrophage, and for dendritic cells. For dendritic cells, we have TLRs and some membrane markers specific for dendritic cell. But how we're going to add those loose nodes? We go back to the literature, and then we identify novel regulatory interactions, and we perform comparison with databases and gene expression. We use ChIP-seq data of 6 histone marks of the monocyte, dendritic cell, and macrophage, and we use chromHMM to annotate 10 chromatin states. At the bottom, I show you a genome browser screenshot of the gene IR4. And according with the color code of the heat map, the only cell type where this gene is active is the dendritic cell. For macrophage and the monocyte, the gene is inactive. And it's according with the biology, because the gene IR4 is the transcription factors that participate in the differentiation of dendritic cell. We select 4 regulatory regions: active gene enhancer, active TSS, poison enhancer, and TSS repress, to search for transcription factor binding sites. The regulatory TF that we use are in the y-axis and the target genes are in the x-axis. The colored square are the binding sites that we found, and the ones with the star are the binding site that was also previously described. We can see that the transcription factor IR4 exhibit several binding sites for our target genes. We also perform RNA-seq analysis for dendritic cell, macrophage, and monocyte, and the genes are clustered according cell type. The blue genes are the specific for dendritic cells, the purple genes are the specific for monocytes, and the orange genes are the specific for macrophages. We can see that the gene IR4 is upregulated into dendritic cells and is downregulated into the macrophage and the monocyte. And this corroborates our findings of the chromatin states. Putting this together, we complete our model, having CSF2 and interleukin-4 as inputs, the 4 signaling cascades, and now our transcription factors exhibit several target genes. The purple nodes are the specific for the monocytes, The orange nodes are the specific for the macrophages, and the blue nodes are the specific for the dendritic cells. We performed several analyses with our model. The first one was to compute the stable states, and we obtained 4. Each of one identifies with a specific cell fate. The heat map, I show you the minimal nodes required to identify those cell fates. In the first column, with any stimulus, the monocyte goes to cell death, and this is also what happened in the laboratory. When we have the stimulus of the interleukin-4, the genes specific for the monocytes are active. When we have CSF2, the genes specific for the macrophages are active. And when we have the combinatory of CSF2 and interleukin-4, the specific genes for dendritic cells are active. We also test some mutations into our model. For example, I show you IR4, and it's required for dendritic cell differentiation. We can see that with the knockout, the only cell fate that is affected is the dendritic cell. MAFB is a transcription factor that is required for macrophage differentiation, and with the knockout, the macrophage is the only cell fate that is lost. We also wanted to know the percentage of the population that can be achieved through time. So in the panel A, I show you that with the initial condition of interleukin-4, almost 100% of the population exhibit the specific markers for monocyte stimulated. With the initial condition of CSF2, almost 100% of the population exhibit the specific markers for macrophage differentiation. And when we have the combinatory of CSF2 and interleukin-4, we see that almost 100% of the population exhibit the specific markers for dendritic cell differentiation. With this, we can recapitulate that our model can correctly identify the final cell fates according each initial condition. I would like to thank to my lab mates and all the people that was involved in this work. Thank you for your attention, and I'm ready to take your questions.